Well, good morning, everyone. Good afternoon and good evening, everyone joining the webinar from around the globe today. My name is Matt Lamb. I'm Director of the Small Molecule Material Science Group at Merck, based in Rahway, New Jersey.
So with all these challenges in mind, we can look at some of the method development considerations. Similar to many analytical technique method development, the first thing we check is selectivity or the ability to discriminate the analyte from the matrix. So in our case, we want to be able to detect the crystalline drug substance to have specific response from the crystalline phase to distinguish from the matrix signals. And if there are more than one crystalline forms or crystalline phases that can potentially be accessible in the dosage form, they should all be assessed in the method. So the method may need to consider multi analyze in a sense. So in the left figure of the PXRD overlays, the peaks marked by the green arrows, they show good selectivity of crystalline drug substance against the amorphous drug substance or against the ASD drug product matrix. So these peaks are good to use for crystallinity characterization. While the peaks that are marked by the red arrows, they have significant overlapping with the drug product matrix and may not be would not be a good signals to use for crystallinity analysis. However, in the right figure of the IR spectra, the peaks of crystalline drug substance marked by the red arrows, they also have significant overlapping with peaks from either amorphous drug substance or from the drug product matrix. And these peaks, although they are not optimum to use for crystallinity analysis, so if we can have a signal with better resolution to the drug product matrix, then we prefer to use those signals. However, in the case where we have no other choice but the overlapping signals, we will potentially leverage advanced data processing techniques such as chemometrics to improve the discrimination ability when use such techniques. Another method development focus point is sensitivity for the crystallinity method. So a typical approach or practice in industry for crystallinity method is to establish a practical limit of detection. This practical limit of detection may not necessarily be the same theoretical limit of detection by the technique, but it is the level of crystallinity that has shown doesn't impact the drug product performance negatively. And this level of crystallinity is able to be detected reliably. So that's what we mean by practical limit of detection. So by using this approach, it does simplify the crystallinity method development effort. So the focus of the method will just be on the failure mode in terms where when there is bioperformance impact by the crystallinity level. There are another note is on the expression of crystallinity limit. So sensitivity can be expressed in as percent crystallinity. However, this percent crystallinity can be expressed in different ways. Either percent of the crystalline drug substance in the total drug substance, including both amorphous and crystalline drug substance, or it can be expressed as percent of crystalline drug substance against the total sample or total matrix. The chart on the left is an example of ASD with 20% drug loading. So within this ASD, if there is 5% crystalline drug substance with regard to the total drug substance, this will correspond to only 1% of crystallinity if we are expressing it as the weight percent against the total sample weight of the ASD. And the chart on the right shows, so if this 20% of ASD is formulated into a drug product at 50% load of ASD, then the same level of crystalline drug substance, 5% of total drug substance compared to 5% of crystalline drug substance against the total drug substance amount, this will correspond to only 0.5% crystallinity if it's expressed as weight percentage of total drug product weight. So this is just something that a little unique for the crystallinity method and expression of crystallinity level that we may need to pay a little more attention. Another consideration is Frank also mentioned that crystallinity can come as from different pathways, can be either sorry. So there are sorry, I'm going to. So other method development considerations. How do I go back? Okay. So as Frank mentioned, that crystallinity can come from different ways, and consideration need to be put in place whether the reference material that is prepared for method development actually represent how the crystallinity is being generated. Because a lot of times the spec-in crystalline drug substance for reference material preparation can have it may be different from the grown crystallinity or residual crystallinity. So the plots on the bottom actually depicts different pathways that crystallinity can be generated. So the plot on the left represent the scenario where the crystallinity is grown from the amorphous matrix, and the crystals in this scenario may have different form, shape, size, or the contact of closeness of contact to the matrix than say the spec-in crystals, which is represented by the middle plot, and it can also be different to the residual crystals from the incomplete amorphization represented by the red plot. So if feasible, the reference material with different crystallinity level should be prepared with similar ways of crystallinity generation to the samples that to be tested. And in these cases, it could be from incomplete amorphization of the ASD or with deliberately grow crystallinity by putting the sample under stressed condition. And if the reference samples are prepared in such ways, then a primary analytical technique will be needed to quantify the crystallinity in these reference samples before they can be used to support method development. That being said, oftentimes than not, that it is not even feasible to prepare crystallinity reference samples in this way with either ground crystallinity or residual crystallinity. So and spec'd-in crystallinity is considered a lot of times a reasonable approximation to the crystallinity observed in the real in the actual samples. So in practice of crystallinity method development, spiking is still the most dominant or most commonly used practical approach for reference sample preparation. So there are a few more sample preparation factors to be considered for crystallinity method development. And this will include both sample preparation for reference sample as well as the actual samples that to be tested for crystallinity. So one factor will be the sample preparation technique, because crystallinity can be lost during sample preparation if it's not properly controlled. So in the plot, the IR plot on the right-hand side, that illustrated the crystallinity loss. So here, it's showing the amorphous peak appearance. And here the peak of the crystalline drug substance signal decrease when the sample is being milled. So that represents amorphous generation as well. And other evidence is that in this peak, there is a peak broadening after a sample is being milled. And so this also indicates that amorphous content is generated compared to the unmilled sample. So, so when the sample is being prepared, we need to be careful not to generate additional amorphous content or alter the amorphous or crystallinity of the sample with the sample preparation technique. Another factor actually we touched upon earlier as well. So particle size of the drug substance or even sometimes the excipients may need to be controlled. A lot of times the reduced particle size can help with control the uniformity of the reference sample preparation, and this will help with the method performance in the long run. And another benefit could be that it can, with reduced particle size, it could reduce preferred orientation that a lot of time may be a significant factor for techniques such as PXRD. And other factors would to be considered can be moisture level in the sample. So moisture level may need to be well controlled as well for techniques that are sensitive to moisture level, such as mid-IR or near IR. So the response we're getting from those techniques are not altered by the moisture level instead of the crystallinity level. So for methods, crystallinity methods, especially when they are entering late phase of development, method validation and transferability also need to be given proper consideration. not like the traditional analytical techniques such as HPLC. Crystallinity method validation is relatively less well established, and so may need sometime it will need special careful design on how the validation should be performed, carried out. A lot of times, we mentioned a little bit earlier as well, so industry often adopt a limit test approach for crystallinity method. And for such kind of a limit test, the validation may be focus on the specificity and a practical limit of the detection validation. For practical limit of detection, validation can be achieved by demonstrating the detection at specific spec level with sufficient signal to noise. And so a lot of times with such kind of a limit test, linearity, like other validation components such as linearity, is not really required. And for methods that need to be transferred into a, say, QC setting or into a production site, other factors will need to be considered. For example, whether the instrument is available and whether the instrument has different configuration, if there is sufficient support of infrastructure, including even the SOPs, how to carry out the test. So these factors all need to be considered in order for the method transfer to be successful. And other things, it happens quite often for some very specialized technique that analysts may not have sufficient expertise to support those testing, even though the instruments is available. And other factors to consider during method transfer would be whether this type of technique has sufficient throughput that it will be required by the crystallinity method testing. So with all these method development challenges and considerations, the ASD crystallinity characterization group recommend the following overall crystallinity control approach. So the first part of the data package included to apply all proper analytical tools available in the development space. This could include those less common, not available techniques in production setting, but they are available in development setting. So with all this technique, we will establish the physical stability envelope below a crystallinity threshold, and this threshold is the one that was that is established, doesn't impact the efficacy adversely. And another component would be to demonstrate crystallinity control below that threshold can be realized by controlling process parameters, storage, and packaging conditions. And so all this data should be generated within the development space. And this, as a combination, would enable that no crystallinity analysis in production is required. So this will be basically the scenario C that Frank touched upon earlier in the ICH guideline slides. There are cases where routine analysis, crystallinity analysis is still required, and typically those will be the cases where development data cannot show that crystallinity is able to be controlled below that threshold without without affecting the bioperformance of the drug product. So in conclusion, in this webinar, we presented unique challenges associated with crystallinity characterization method. And we also provided guidance for crystallinity characterization best practices. So this is to promote consistency across industry. The ASD crystallinity characterization working group advocated to the industry. Again, just to re-emphasize, we want to apply all common advanced and emerging analytical techniques in the development space to define crystallinity failure modes. And we also want to emphasize to develop appropriate manufacturing, storage, and packaging control strategies to control crystallinity. And so with this extensive development work will be able to enable no crystallinity test in the QC setting. So basically, even whenever possible, the scenario C will be encouraged to the industry. As mentioned earlier, this webinar is based on the white paper that was published by the ASD crystallinity characterization working group. And so we thank all the authors for the white paper. And we also thank the current member of the working group who represent their individual pharma company for helpful discussions with preparation of this webinar. And with that, we are ready to take questions with our panelists.
Okay, thanks, Frank and Lily. Hopefully everyone has a good appreciation of certainly the challenges with developing these physical characterization methods and the recommendations of the working group. We're going to go ahead and take some questions, and we're going to bring some others onto the panel from the working group. So I'd like to just quickly introduce Sai Chamarthy. Sai has a master's from Duquesne University in pharmaceutics, specializing in drug delivery systems for DNA-based vaccines, and a PhD from Purdue in pharmaceutics, specializing in mechanistic understanding of functionality in pharmaceutical materials. Sai is currently director at Amgen, responsible for commercialization of synthetic molecules. We also have Ashish Punya from Merck. Ashish has a PhD in polymer chemistry from the City University of New York. Over 8 years experience at Merck in small molecule material science and implant formulation development, and is currently principal scientist in the Analytical Enabling Capabilities Group at Merck. And finally, Paul Stroud. Paul has a PhD from the University of Southern Mississippi in polymer science and engineering, 15 years at Eli Lilly and Company in the synthetic molecule design and development, and is currently senior director in material science and physical characterization. I'm going to try and navigate the questions here as I switch. Okay, so while I'm getting set up here, maybe Lily, I'll start with a question from you. Can you talk about maybe some of the differences in how you might approach characterizing the amorphous solid dispersion itself versus when it's formulated in the drug product?
So the difference in characterizing the ASD versus the characterizing the crystallinity when the ASD is formulated in drug product?
Yeah, if there's any unique challenges or differences in approaches you might take.
Yeah, definitely. So I think it's basically similar challenges as we touched upon in the webinar. So specificity and the sensitivity can be quite different from in the ASD versus in the drug product. Typically in ASD, the matrix is a lot more simple than in a drug product. The drug product has a lot more excipients and with crystalline signals. So that can present a challenge with specificity. And inherently, when you put the ASD into a drug product, you are diluting your sample concentration even further. So the same amount of crystalline drug substance where you may be able to detect in the ASD, but when you put it in a drug product, it's at a much lower absolute concentration, and so you may be facing detection limit challenge in the drug product. So these are the typical two considerations, but other consideration would also include like particle size control, moisture level control. It may be more challenging in the drug product than say for the ASD alone. So
Okay, yeah, thanks so much. I'm looking through some of the questions here. Again, folks online, go ahead and put your questions in the chat. I'd also just make a plug that this is the first webinar that we've done as this working group. So we're kind of working things out with the technology and the presentations, but if you have suggestions of other webinar topics that you'd like this group to tackle in the future, go ahead and put those in the chat. And as a working group, we can work to try and get that content for you. Hopefully you saw with the members and the representations across the industry, a lot of experience, a lot of expertise in this presentation and in the working group. And so we certainly love to leverage that for the benefit of other webinars and education across the industry. Let's see, Sai, there was a question that says, what type of data package is needed to justify the scenario C, no testing? And has the working group done any survey or case study collection on this point? I'll quickly just comment on the survey case study. So the working group right now is in the middle of doing some benchmarking surveys around almost all aspects of amorphous solid dispersion, both formulation, development, and characterization. And so next year we hope to have the output of that in a few different ways. But I'm going to go ahead and go back to the slide that they're referring to. But maybe Sai, if you want to speak to control strategies and in your experience, what kind of data packages might be needed for scenario C where we wouldn't have a control test?
Yeah, so scenario C essentially leverages the development data that you would generate and present in your process development and formulation development section, right? And in that particular section, you would demonstrate that a known amount of crystallinity that is above the crystallinity that is generally seen in your drug product through the light, through its shelf life, does not affect the QC method. And one additional step could be to show that there is no effect on bioperformance, which can be achieved through a PK study. Right? Once you have established those two steps, then essentially you do not need to control for crystallinity in the drug product or ASD on a routine basis.
Okay. Thanks for that, Sai. Frank, I'll maybe get your perspective as well. Do you have, can you speak to your experience, where you've been involved maybe successfully in getting that scenario and what approaches did you take to have no control test required?
Yeah, well, usually it also depends what you are comfortable as an organization to present to regulatory agencies. I'm not sure if I remember any case where we at least did not put crystallinity to be monitored and had an XRPD method just to make sure and know that we can go through with that at the agencies.
Thanks, Paul. Would you say there's one technique that you find you use more routinely than others? We heard a lot of different techniques today, and some obviously have their strengths and challenges, but is there one that you think is, you tend to see to use more and more in development for characterizing crystallinity?
Yeah, certainly. X-ray powder diffraction is the gold standard, in my opinion, as far as detecting crystalline materials, especially if a lot of your matrix is amorphous. So, you get outstanding specificity and very good sensitivity. But, I think the backup after X-ray is going to be a lot of the spectroscopic techniques as well. Those are very common, and both of those are usually capable of being transferred to a QC environment. But obviously, the more sophisticated techniques would be used as an orthogonal method to support the more common techniques.
Okay, thanks. We got a question about a specific slide on the sensitivity around inactive components, what is meant by ASD and non-ASD. So Lily, since you presented this one, maybe just briefly talk about what we're trying to show here as far as sensitivity and essentially dilution effects from the drug product.
Can you clarify that question a little more?
So the question says, didn't really understand the sensitivity concept. Could you please elaborate a bit on inactive components and what is meant by ASD and non-ASD? So here we talk about inactive components not being non-ASD, inactive components as part of the ASD.
Okay, so maybe some people are familiar with the expression of excipient and versus drug substance. I think the inactive components are the excipients that are not part of the drug substance. So an active components will be just the drug substance, either in amorphous form or in crystalline form. So this is what the inactive components is meant. So in ASD, the inactive components will be the polymer to make the ASD, but in drug product, it will be the polymer in addition to other drug product excipients for the ASD drug product. Yeah.
Great, thanks for clarifying. Yeah, and just to reiterate for folks that may not be aware, the amorphous solid dispersions generally contain both the drug and some kind of polymer or other active ingredient, most typically to stabilize it in the amorphous state. So I think they're trying to call out here your amorphous solid dispersion may only itself be 20% drug loading, something like that, and then that gets further diluted into a tablet or a formulation. Okay, got a lot of questions here. I'll try and go through them, but maybe in the meantime, Ashish, one of the topics that came up was around, making your standard preparation, if you're spiking it or you're using grown-in material to make those standards to develop your method. Can you talk a little bit about blending and preparing those standards for sort of method robustness and some of your experience?
Yes, there can be a few methods, lab scale methods that can be used. One approach can be like a shaker mixer, like a Tarula mixer, that can also be used in a lab scale to mix these. And homogeneity of these calibration samples, those are critical, as also evident from the presentation that we just had. Other option can be like an acoustic mixers. Those are usually available in development labs and pharmaceutical industry. And then that can be a second method, like mortar pestle mixing. That is also still widely used, and we can still obtain homogeneous samples. So yeah, I think there should be a lot of focus on appropriately obtaining homogeneity levels in these calibration samples, and we have to assess carefully those approaches. So sometimes it's a mixture of these approaches that the development scientist has to utilize.
Okay, thanks, Ashish. So given we can go over 15 minutes or so if we like. I think maybe in interest of everyone's time, maybe a few more questions, and we'll let others get on with their day. But let's see. So there was a question about the white paper, the link. So I don't know if I can put this in the chat. Maybe I can. But here it is. You can obviously punch this into. Okay, that's probably not working. So Go ahead and write it down. I'll leave it up here. You can also obviously search in Google Scholar based on it. This was, again, the white paper that the members of pretty much the original working group had published and came out last year, and was most of the content that we presented today. As a working group, I think there's a question of what else we're doing. As I mentioned, we're really interested in benchmarking across companies, particularly now that the amorphous solid dispersion is more mature and routine in the industry, and we'd like to see actually what companies are doing most typically, and so we'll be looking for other webinars and potentially publications on the output of that once we get through that benchmarking survey within the IQ. Let's see. Frank, given your background in spectroscopy, can you talk about maybe developing chemometric methods with spectroscopic techniques and the challenges or benefits of doing that?
Most of the time, the biggest challenge is to actually determine specificity in spectroscopic methods. Usually, you would I think show specificity by, or what people would like to show the specificity is that you have the spectra of the crystalline drug substance and then have a response from your chemometric model that is an exact overlap of the crystalline drug substance spectrum and the response of the chemometric model. If we are talking about ASDs and spectroscopy, you will not only interrogate the gain in crystallinity in most of the cases, but also the loss in the sample of amorphicity, and also interactions between the polymer and the API that are changing with growing crystalline content. And this resulting effect was translated in the chemometric model to a loading, which in principle corresponds to a spectral signal. And this will, in most cases, be not exactly the same than the crystalline content. So this is usually a challenge that you get afterwards when you got your model in discussing about it. What is another practical challenge I can talk about is the transferability of model parameters from chemometric software to chemometric software. Instruments usually operate with their own chemometric suite in the software that they are running with, or they might have a different chemometric engine than where you did your method development, or where you could comfortably do your method development. And this can lead to difficulties when you want to transfer a method from that you have developed on a bigger chemometrics package to then an instrument software package, as certain, for instance, pre-processing routines will not be available, or areas are truncated differently and will lead to slightly different results.
Great, thanks. There's a question, and I guess I'll open this up for anyone on the panel around using total X-ray diffraction for characterization. Anybody have any experience using total X-ray diffraction as a tool for detecting crystallinity? Is it widely used? That's basically the question.
Yeah, I'm not quite sure what they mean by total X-ray diffraction. I'm not sure if that's where you take all of the crystalline peaks and account for the area of those. I personally have not utilized that. I know that it's available in some of the software packages associated with the instruments, but I think that it's something that if it works for you and you have the ability to, back the data, I don't see this being an issue to potentially try.
Mm-hmm. And just to follow up on that, Paul, there was some discussion in the presentation a little bit about whether you're actually developing quantitative methods or limit tests. Do you find for X-ray diffraction, in your experience, that you're typically actually making quantitative methods, or are you generally doing more of a limit type test?
I think it really depends on your amorphous solid dispersion system. If you have a system that is very unlikely to ever nucleate and grow, I think a limit approach is very acceptable. However, when you do have a formulation that might be a little more susceptible to nucleate and grow actually in the formulation and in the drug product, then I think quantitation, at least in the development area, would be recommended to be able to do that so that you can follow and understand how much crystalline growth you might see during stability or stress stability studies.
Excellent, thank you.
And can I get a quick point on that as well? Like a general concern from the physical stability perspective is the first observation of amorphous to crystalline form conversion. In that case, the limit tests are really appropriate in those. Like subsequent crystal growth, in a number of cases, that is not the major concern, and that's why the limit test, the physical stability, is generally even like more suitable in these cases. Yep.
So during development, Ashish, you're saying that we're developing limit tests so that as we're maybe stressing the amorphous solid dispersion, we're looking to see when, at what time point, at what condition we're starting to see it. And so, you know, a limit test is usually sufficient. I think certainly in the QC space, if we ever have to get that, you know, for whatever reason we think that we can't control it, and we're going to be having to do the tests, you know, later stage development, QC, things like that. There may be some expectation of more quantitation, and I think in our experience, that's some of the nuance and the difference there. But thanks both for weighing in on that.
Can I add one point on the total XRPD
Absolutely.
considerations, right? So I think if you're looking at an amorphous solid dispersion alone, where you just have the polymer and your amorphous API, then it probably becomes easier for you to do a total XRPD and try to quantify. But in the drug product, because of all the other components, it's generally not recommended to do that. It is easier to identify a peak of interest where you essentially don't have any interference, and then that gives you better sensitivity. Yeah.
Yep. All right. Great. Thanks. So I think we got through most of, if not all, the questions. We'll try and follow up if we can. I want to thank the speakers today, Frank and Lily. I want to thank the other panelists that joined from the working group. I want to thank the IQ for obviously putting this framework together that allows us to come in this pre-competitive space and collaborate in this consortium, and also sponsoring the webinar. Thanks for everyone today for joining. Thanks for your questions, engagement. Hopefully you found this interesting. I think we did get some suggestions on other future webinar presentations, so we'll look to see what we can do there. And if you're interested in participating again in the working group, and you're already from a company that's a member of IQ, go ahead and connect with your IQ liaisons within your company to see if you can get involved. And you know, we'd certainly love to have you contribute to the objectives of the team. So with that, I'm going to go ahead and close. Thanks everyone. Malaya, any other final comments from the IQ side?
No, nothing further.
Okay, thanks very much. We'll go ahead and stop it here, and yeah, enjoy the rest of your day.
Everyone.
Thank you.
Thanks.